Hey everybody, hope everyone's doing well today. Hey everybody, hope everyone's doing well today. Hey everybody, so we are doing well today. Hey everybody, so we are doing well today. Hey everybody, going crazy. Okay, looks like it's connecting to the chat, so hopefully I can see what you guys are saying. But today we are going to be going over cellular respiration. Awesome. You guys should have access to these worksheets. They might be on your D2L page, and you might have to check with your professor about that because it might be different per class. But I'll go ahead and get started. Just trying to see if I can see a chat.
You're at the mercy.
Okay, awesome. Yes, I can see you. Great. I am seeing highs. Awesome. Thank you. Okay. So the first one says, how can cells convert the energy in glucose to ATP? So we have this really nice diagram. It has an overall simplified down version of glycolysis into pyruvate oxidation into the Krebs, or it's also called citric acid cycle, and then into oxidative phosphorylation. So the table that we'll be starting with asks where each of the reactants are in the overall process. So this first one that we're starting with right here is also known as glucose. And as we can see here, that's one of our reactants. So, oh, sorry, it looks like it's not capturing what, oh, great, okay. I had no technical difficulties when nobody was watching, of course. Awesome. Oh, the extra credit, sorry. I did not address this yet. So I have a Google form that I'm going to send into the chat at the end of the live stream, and what you'll do is you'll put in your name and your email, and then you'll also put in the date that we viewed the live stream today, and that's how I'll keep track of your attendance, and then I'll send that into your professors, and they'll handle it from there. So, does that sound good? Awesome. Okay, so we're starting off with glucose. So if we look up to this blue box, we can see glucose right over here. So we can say that glucose is found in glycolysis. It always trips me up that there are two Y's in glycolysis, and I always forget. Okay, I will also try to, here's what I'll do. I'll send in I'll make a note of it, and then I'll put in the link in halfway through. Is that okay? Will that give you enough time? Just let me know in the chat, but I'll try to send it in by 5:30. Okay, so moving on to, no problem. Moving on to oxygen. So we can also see oxygen here as one of our reactants. However, what's interesting about oxygen is that it skips glycolysis, pyruvate oxidation, citric acid cycle, and is actually only used in oxidative phosphorylation. Oh no. Sorry. Okay. Oxidative. Long word. Awesome. Okay. The video is not playing for you guys. Okay. That is strange. looks like, okay, sorry, let me write hello up here. Yeah, it's a bit laggy for sure. I'm going to keep ahead and going, and I'll try to slow down with it. Thank you guys for bringing this to my attention. This is the first time that I'm getting to do this with a live audience, so I appreciate your guys' patience and everything. Okay, so for carbon dioxide, that's the first product that we're seeing on the product side. Where carbon dioxide is found is in pyruvate oxidation, right over there, and it's also seen in the Krebs or citric acid cycle. And Krebs. Moving on to water for our products. Water is found only in oxidative phosphorylation once again. And I also am sorry that this is kind of laggy today. I will definitely try to figure this out by next week. Just so you guys know that this is recorded, this is also put on YouTube. It might have a clearer recording that way. The stream platform that I'm streaming it on is also normal, and this hasn't happened before, so I am wondering if it is Google as well. Awesome. And then for energy, which is the most important part of all of this, we have a couple of things going on. So I'm going to start going through glycolysis, kind of through those processes, and then listing what we generate. And this diagram gives us a good idea of what that will look like. So from glycolysis, which happens 2 times, we generate ATP, and then from the Krebs cycle, I know that what this says up here, but we generate 2 GTP, and then an oxidative phosphorylation, which I'm just going to abbreviate and put OXP. We can generate up to 34 ATP. So oxidative phosphorylation is really our money maker when it comes to energy, and that's where within these processes we're going to generate the most. Attendance will be sent at 5:30. You're going to fill in a Google form, and then from there that information will be passed on to your professors. And yes, 2 ATP in glycolysis, I should clarify this. So glycolysis happens in a cycle of 2, and that's why 2 ATP are generated. Okay, so for question 2, it says, in cellular respiration, the oxidation of glucose is carried out in a controlled series of reactions. At each step or reaction in the sequence, a small amount of the total energy is released. Some of this energy is lost as heat. The rest is converted to other forms that can be used by the cell to drive or fuel coupled endergonic reactions to make ATP. Last week we identified endergonic as energy consuming reactions, which means that this energy can be used to build things or can be used to make ATP, the energy that's released. So the first box asks what the overall function is of glycolysis or the multiple functions. So what happens in glycolysis specifically is that glucose gets oxidated into 2-pyruvate. So that's one thing that happens. And then the second thing that happens is that it also generates 2 ATP and 2 NADH per glucose. So for every 1 glucose, it generates 2 ATP and 2 NADH. The second box asks what the overall function of the Krebs cycle is, and that is, 1, the oxidation of pyruvate / acetyl-CoA to CO2. I also wonder if removing my face might help this. So I'm going to make this bigger. There we go. This might be better. Okay. 2 CO2. Once again, thank you guys for your patience. And 2, this the Krebs cycle generates 2 GTP, 6 NADH, and 2 FADH2 per glucose. So generates 2 GTP, 6 NADH, and 2 FADH. And then finally, what the overall functions of oxidative phosphorylation is that it oxidizes NADH and FADH2 to water. To H2O, or you can, oh, it's fine if you're late. You can write this down while we're going. I am under the assumption that the keys to this will also be posted after. So if you would like to just sit and absorb, that is totally okay. We do encourage you. There are blank worksheets that you can work through this with me too, just for muscle memory and yeah, for the future. And you will fill in something at 5:30. I'm going to send in an extra credit link while you'll fill in your information for attendance then. Okay, so this can also be not only converted to H2O, but it can be regenerated into NAD and FAD. And here this generates H+ ions. So it creates a chemical gradient which drives ATP synthase and can therefore generate a bunch of ATP. For gradient. And we always say like approximately 38 ATP. It's kind of APP. No. kind of the golden number, ATP. Okay, awesome. And if you guys have any questions, of course, just feel free to ask in the chat. So this number 3 kind of has a typo. There was supposed to be a chart here, but I'll be able to go through with you I'll go through it with you a little bit. So what it's asking is, it's going through products and reactants and asking whether it's used or produced in either glycolysis, the Krebs cycle, or oxidative phosphorylation. So I'm just going to put glycolysis up here, Krebs up here. I know I'm writing over the question just for more room. And then oxidative phosphorylation. So it's asking about glucose, which gets used in glycolysis and also used in oxidative phosphorylation. It's asking about carbon dioxide and where it gets used and produced. And carbon dioxide is not used in either glycolysis, Krebs, or oxidative phosphorylation. It is only seen produced in the Krebs cycle. And I'll use dashes so that we can see this better. ATP is produced here in the form of GTP, and also in oxidative phosphorylation. ADP and PI are both produced and used in glycolysis. It is only used in the Krebs cycle, and it is again used in oxidative phosphorylation. Okay, now I'm kind of losing room. I'm just going to write up here. So we know that this glycolysis, for NADH, in glycolysis up here, it is produced. So the difference between ATP, and yes, I can see your chat message. So the difference between ATP and GTP, it's not much. So you know the base pairs and nucleotides, we have A, T, G, and C. So these are actually, they can be attached to phosphate groups. So ATP stands for adenosine triphosphate, which sounds a lot like adenine. And GTP is basically guanine triphosphate. So it just, the G or A just constitutes what base pair is attached to it. You'll primarily see ATP, GTP, and UDP used, but you won't really see TTP or CTP. But that's all that really means. It's not, I wouldn't get too hung up on it. It's just an energy source, because breaking a phosphorylation bond is really high energy, and that's what yields so much of it when we are generating ATP. So I hope that helps that question. NADH is also produced in the Krebs cycle, and it is used in oxidative phosphorylation. And then last but not least, NAD+ is used in glycolysis and in the Krebs, but it is produced in oxidative phosphorylation. I know that was a bit messy, so. Okay, number 4. The cell supply of ADP plus PI and NAD+ is finite or limited. What happens to cellular respiration when all the cell's NAD+ has been converted to NADH? So there's a lot of processes within this that require the conversion of NAD plus to NADH. Both glycolysis and the Krebs cycle produce NADH. So as we saw up here, NAD plus is used in both glycolysis and the Krebs. So if neither of those can be used, then both of those processes will cease to function. So we can say if there is no NAD plus, glycolysis and so if there's no NAD plus, glycolysis in the Krebs cycle cannot proceed and will shut down. Number 5. If the Krebs cycle does not require oxygen, why does cellular respiration stop after glycolysis when no oxygen is present? So this usually can trip up students. It was very hard for me to understand this. But when no oxygen is present, oxidative phosphorylation can't occur. And so all of the NADH that's produced in glycolysis and the Krebs cycle can't be regenerated into NAD plus. And so when there's no NAD plus, like we said in answer 4, pyruvate can't be converted into acetyl-CoA that now can't go into the Krebs cycle, which can't fuel oxidative phosphorylation. So I will write that down in simpler words. Okay, therefore NAD plus will not be restored. And I'm going to just draw an arrow to 4 because we kind of answered that and why that's the case in number 4. Okay, and I know this is kind of split on 2 pages, so I'm just going to read it and then we'll answer it in the box. And I will actually make it so that you can see it. Okay, so many organisms withstand periods of oxygen debt, anaerobic conditions. Yeast undergoing oxygen debt converts pyruvic acid to ethanol and carbon dioxide. Animals undergoing oxygen debt convert pyruvic acid to lactic acid. Pyruvic acid is fairly non-toxic even in high concentrations. Both ethanol and lactic acid are toxic even in moderate concentrations. Explain why this conversion occurs in organisms. So, when no, I know that we've said this a few times, but when no NAD plus is available, glycolysis stops and no ATP will be reduced. So in that case, the cell would die. The conversion of pyruvic acid to lactic acid or ethanol requires the input of NADH and generates a NAD+. So though fermentation can seem like more harm than good because lactic acid can be toxic in high concentrations, it pumps out a lot of NAD+, which can further push glycolysis and the Krebs cycle and produce more ATP. So fermentation can produce ATP on its own at a modest amount at 2 per. So I will, write this down. Awesome. So the main message is that if there's no ATP, of course, the cell dies and there's no function. And fermentation can regenerate NAD+, and also can provide a small amount of energy for that cell to keep going. I know that it's not 5:30 yet, but just to focus on the next question, I'm going to go ahead and send a link in the chat. Oh, actually, that is the longest URL. There are 2 links, and I think they're both the same, but one is a shorter link, so I hope that that's okay. But just go ahead and fill that out, and awesome. Okay, moving on to 7. How efficient is fermentation? How efficient is cellular respiration? Remember that efficiency is the amount of useful energy as ATP gained during the process divided by the total amount of energy available in glucose. Use 686 kcal as a total energy available in 1 mole of glucose and 8 kcal as a total energy available in 1 mole of ATP. So if we're testing efficiency of fermentation, we can do this mathematically by using these values to really show how great of a difference there is. So for efficiency of fermentation, we know that there are 2 ATP generated in fermentation. And so using this 8 kcal as energy per 1 mole of ATP, multiplied by 2 ATP will give us 16 total kcal, because these cancel. And in terms of the percentage of per mole of glucose, we can say 16 kcal divided by 2 moles of ATP, because we're getting back to that 8 per 1 mole of ATP. And this is divided by 686 kcal per mole of glucose. And this brings us to around 2.3%. And if we look at the efficiency of aerobic respiration, we can do that same conversion that we did for fermentation, but we generate approximately 38 ATP. Yes, it's kilocalories. Thank you for answering that. Which is a lot more. So we're going to multiply this by 38 instead. And that is about 304 kcal. Yes, the attendance sheet should be open. I can send it again. Sorry, just 1 second. Send. Apologies for my crazy loud mechanical keyboard. Okay. Three and 4 kcal. So we're just going to do that conversion, and we're doing 8 kcal per mole of 1 ATP. And we're going to divide that. Oh, shoot. No, we're using 304. Okay, so, sorry. 304 kcal per 38 mole over 686 kcal per mole glucose. And this percentage comes out to 44.3%, which is a lot greater than that of the efficiency of fermentation. So we can see here why we definitely prefer aerobic respiration over those like fermentation. So moving on to 8, why can't cells store large quantities of ATP? Hint, consider both the chemical stability of the molecule and cell's osmotic potential. In general, cells really like to keep things flowing in and out. They don't like too much stuff within, and they don't like a bunch of stuff out either. But one of the issues with storing ATP is that the osmotic potential would change. So this is why cells can't store things like glucose either. So in the normal body temperatures, it's like I said, it's just difficult for them to store anything at any time. And so because ATP is so small, like having a lot of ATP within a cell will make it hypertonic to its environment, meaning that there's more things in than there are out. If that was the case, then the osmotic potential would want to rush into the cell, and it could cause the cell to burst, and so there wouldn't really be use for that. So those are a couple reasons why the cell can't produce high amounts of ATP. So would change osmotic potential. So I think, yeah, and I just saw this too. And the key, I think what was happening was that, I don't know if it wanted to divide it by 2, but just say that it was per 2. So maybe, I think it's just 16 out of 686, and then 304 divided by 686. Sorry. And yes, usually, I mean, ATP is relatively small and likes it and has a hard time traveling through that hydrophobic part of the membrane. And so it usually requires gradients and things like channels in order for it to cross. So that is correct, yes. And cell could potentially burst. Great. Given that cells can't store ATP for long periods of time, how do they store energy? So because energy is used pretty rapidly, things that we store it in are like larger molecules. We can store them as fats, we can store them as starches, we can store them as oils. And this would be one of those examples of an endergonic and anabolic reaction. And for part C, what are the advantages of storing these alternate forms? So building these larger molecules has a reduced effect on osmotic potential. So we said earlier that ATP is kind of a tricky molecule. We don't want to store too much of it because it would change the potential within a cell. But having larger molecules, it's harder for them to move around. They can't do a bunch. And so that's one positive. And they also are a lot more chemically stable than ATP. Generally, ATP likes to do what it's best at and release energy for things. And when we're building things into more stable molecules that are less mobile, then they're easier to store. And then more stable. Great. So for number 9, which is once again at the bottom here, I'm going to read half of it, then we'll finish it on the other page. So to make a 5 molar solution of hydrochloric acid, we add 400 milliliters of 12.5 molar hydrochloric acid. Oh my gosh. To make a 5 molar, so we're making a 5 molar solution of hydrochloric acid, we would add 400 milliliters of 12.5 molarity to 600 milliliters of distilled water. Before adding the acid, we place the flask containing the distilled water into the sink because the solution can heat up so rapidly that the flask breaks. How is this reaction similar to what happens in chemiosmosis, and how is it different? So to go over what chemiosmosis is, just a refresher, chemiosmosis is a chemical ion gradient that is also within like water. So we can say chemiosmosis. So osmosis having to do with the movement of particles with water, and then chemi as in chemical or charged. So some similarities in chemiosmosis that we're seeing here in this solution, we're generating a difference in the ion concentration gradient. So in chemiosmosis, there's that change, and when we're putting acid into water, we're also making a change in that H plus ion gradient. Acids tend to have a lot of H plus protons, and so adding an acid into a water is going to change it and affect it in that way. And we could also say this gradient will flow down and generate heat. And then some differences between the two is that chemiosmosis sets up, I'm going to write chemiosmosis, sets up the H plus ion concentration gradient, but the energy release is controlled as it's moving through the ATP synthase. So it's not just bursting into heat or else we would die. So it passes through the ATP synthase, and then that is what generates that enzyme. So ATP is generated. So I will say that some energy is lost as heat. So it's not that none of it is, but a lot of it is captured in the chemical bonds of ATP, and there's not nearly enough to combust as it would in that chemical reaction. So to answer that question in the chat about what happens when the acid solution gets mixed with water, I know I really like chemistry, so I always try to bring it in, and I'm sorry if that makes it confusing. So basically, we define something acidic as having a high concentration in H plus ions. And so something like hydrochloric acid getting mixed with water, this has a lot more, oh awesome, yay. So this hydrochloric acid has a lot higher of hydrogen ion concentration than something that is relatively neutral like water. We know that water has polar properties to it, of course, but in terms of the difference between that and hydrochloric acid is that it has a lower pH and a larger pKa. So when you add a bunch of, like if you have a pitcher of water, and let's say that's water, if you add a bunch of H plus ions, this is decreasing the pH of this, increasing the pKa, and therefore it's making it have more of an ion gradient. There's more charge there. There's a lot more going on than there was with just water. So I hope that helps with this. So basically, when we're relating it to chemiosmosis and we're talking about this big ion gradient and all of these protons moving in and out, highly acidic solutions have a lot of protons, and that's why we can relate the two pretty well. But the energy that's released from that reaction, yeah, of course, the energy that's released from that reaction is all heat. But in a cell, it's a lot more controlled than that. We use it for ATP, and so, yeah, I think I said that like 4 times. I'm sorry, you guys, I'm like rambling now. But yeah, chemiosmosis is really important, generates a lot of our energy. So now we're going to move on to cellular respiration and experimental scenarios. So this is more taking what we know and kind of using it for questions. Yes, exactly. Exactly. So it's like if you're thinking about different pathways that a bunch of energy can go, and it's like through 3 doors, and one of them's heat, one of them's ATP, and the other one's to make candy or something, like exactly. We're just going to take that energy and put it into making ATP instead of just having it get off as heat. That was a perfect way to write that. I don't know why I keep rambling. I'm so sorry. But yes, exactly what you said is correct. Okay, so if it takes 1,000 grams of glucose to grow 10 grams of an anaerobic bacterium, how many grams of glucose, thank you, would it take to grow 10 grams of that same bacterium if it was respiring aerobically? So that's with oxygen. Estimate your answer. So for example, if it takes X amount of glucose to grow 10 grams of anaerobic bacteria, what factor would you have to multiply or divide X by to grow 10 grams of the same bacteria aerobically? So this is kind of wordy, but we can just, once we kind of take out the parts, it'll make a lot more sense. So aerobic respiration, we said with oxygen, has a max of 38 ATP. And this is per glucose. Anaerobic respiration produces a max of 2 ATP per glucose. So as a result, if we're looking at this to this, we could say that aerobic respiration is 9 times more efficient. Nineteen times more efficient. So in that case, we would need, on the flip side, 19 times less glucose if respiring aerobically. So even though these could have the same input of glucose, this can make 19 times more than that. So it would only really need 19 times less to make just as much. So if there's 1,000 grams of glucose, we can divide it by this number 19 and then get approximately 50 grams of glucose. And this is just saying that there are 50 grams of glucose required if respiration is aerobic. Does this make sense? Okay, cool. So B, mitochondria isolated, kind of. Okay, I want to hear yeses. I'm going to go over it one more time. So this is with O2, this is no oxygen. So with aerobic respiration, we can make 38 ATP molecules. Anaerobic, we can only make 2. From this calculation, what we say is we can deduce that it's 19 times more efficient. I think that's correct. Yes. So we're just saying that, like, for every one, this one can produce 19. 38 divided by 2 is equal to 19. I think I should have said that first. I just kept going. So 38 divided by 2 is 19, and that's why this one is 19 times more efficient. So therefore, if we have 1,000 grams of glucose, if we know that we can produce something 19 times more than, we're going to divide that by and extrapolate from that the amount of glucose required. I know that this is kind of difficult. It's kind of a roundabout way of saying, like, if anaerobic requires 1,000 grams of glucose, then aerobic would only require 50. So, like, this would both generate the same amount of ATP. I feel like I didn't explain this well, so I'm just going to try to do the math like one more time. So we're saying up here that it takes 1,000 grams of glucose to grow 10 grams of anaerobic bacterium. Anaerobic means without O2. So we started with anaerobic, and now we're finding aerobic. It took 1,000 grams of glucose to grow 10 grams. So we're seeing how many grams of glucose would we need to grow the same amount in an aerobic condition. In anaerobic, we generate 2 ATP. In aerobic, we generate 38 ATP. To see how much more efficient this is, 38 divided by 2 equals 19. So in that case, if we did 1 ATP here and 19 here, we could say that for 1 ATP molecule generated in anaerobic, there are 19 generated in aerobic. That's why it's 19 times more efficient. I know that I didn't really explain this well, so thank you for having me just do it one more time. So when we're having 1,000 grams of glucose, we're just going to divide that by 19, which is how much more it's efficient by, and that's how we get 50 grams. So I hope that helps. Awesome. Okay. B. Mitochondria isolated from liver cells can be used to study the rate of electron transport in response to a variety of chemicals. The rate of electron transport is measured as the rate of disappearance of O2 from the solution using an oxygen-sensitive electrode. How can we justify using the disappearance of oxygen from the solution as a measure of electron transport. So we can start by just simplifying and writing down the equation for aerobic respiration. And I'm just putting E there for energy. So the final product produced is 38 ATP. And there are 6 moles of oxygen consumed. But how we can explain the fact that the disappearance of oxygen, because even with this, we can predict that 38 ATP is going to be produced from this. And even though there are 6 molecules of oxygen consumed, we still on this side expect 38 ATP. And I guess I shouldn't have written it this way because that looks like it's products and reactants. But basically, so for every 6 oxygen molecules consumed, we expect 38 molecules of ATP to be produced. And yes, O2 is the last molecule in the transport chain. That is also correct, yes. Okay. Humans oxidize glucose in the presence of oxygen. For each mole of glucose oxidized, about 686 kcal of energy is released. This is true whether the mole of glucose is oxidized in human cells or burned in the air. A calorie is the amount of energy required to raise the temperature of 1 gram of water by 1 degree Celsius. So that is it converted into calories. The average human requires about 2,000 kcal of energy per day, which is equivalent to 3 moles of glucose per day. Given this, why don't humans spontaneously combust? And the answer for this is on the next page again. And I know that this is going a little bit over time. This might go a little bit over time, so if you have things to do, of course, I appreciate your guys' patience and that this took a little longer because of technical difficulties. So we can say for this, as we said earlier in question 9, the energy from oxidation of glucose is not released all at once in burning. So we get some ATP from glycolysis, we get some ATP from the Krebs cycle, and we get some ATP from the electron transport chain. And in each of these, it releases a small amount of energy stored. And so we can say that some of this energy is captured as NADH, FADH2, it's captured as ATP, it's captured as GTP, and we can still say some is lost as heat, but of course the heat loss does not occur all at once, just as generating ATP doesn't always occur all at once. Okay. A gene has recently been identified that encodes for a protein that increases longevity in mice. To function in increasing longevity, this requires a high ratio of NAD plus NADH. Researchers have used this as evidence to support the calorie restriction hypothesis for longevity, that a decrease in total calorie intake increases longevity. How does this requirement for a high NAD plus NADH ratio support the caloric restriction hypothesis? So when we eat less stuff, we can say, if we eat less stuff or decrease our calorie intake, we're also decreasing the rate of glycolysis in the Krebs cycle because there's less to work with. So if there's less working glycolysis in the Krebs cycle, there's going to be less NADH produced by glycolysis. So the NAD plus NADH ratio will increase because we're not using as much NAD plus. So we can also say if there's a decrease in use of glycolysis in Krebs, there's an increase in NAD plus concentration. So therefore, the NAD plus NADH ratio will increase. Awesome. An active college age athlete can burn more than 3,000 kcal per day in exercise. If conversion of 1 mole of ATP to ADP plus PI releases about 7.3 kcal, roughly speaking, how many moles of ATP need to be produced per day for this energy need to be met? So we said 3,000 kcals per day, and then conversion of 1 mole releases 7.3 kcal. So we can say for this, thank you so much, no problem. Kcal per day divided by 7.3 kcal per mole of ATP is equal to 411 mole ATP. Awesome. If the molecular weight of ATP is 573, how much would the required ATP weight in... If the molecular weight of ATP is 537, how much would the required ATP weight in kilograms? We got 400 mole up here because we started, it said 3,000 kcals per day in exercise, and that there are 1 mole of ATP to ADP released in 7.3 kcal. So when we're dividing these two, kcal cancels out. And I know this is 3,000 kcal per day, but it's also just asking, yes. So yeah, that's a better way to write this. What we're really asking is just 3,000 divided by 7.3, and that's going to get you the moles of ATP. So in this one, this is just asking us to convert ask how much weight is in this many moles. So the weight of ATP is 573 grams per mole. And then we're going to multiply this by the amount of moles that we found in the previous problem, which was 411. This is equal to a pretty large number, about 235,503 grams. Or if we were to convert it into kilograms, it would just be 235 kg. Yeah, of course, no problem. Thank you, guys. Okay, last question. Explain these results. So, like, basically, how can this person exercise so much, require so many calories, and still have ATP? Is we can say that ATP is broken down to ADP and PI, and then regenerated into ATP. Awesome. So this brings us to the end of the stream, everybody. Thank you guys so much for coming and bearing with me. I know this is super laggy. Oh my gosh. I will fix this next week. Yeah, so thank you guys for filling out the chat. I will definitely make sure to keep record of that. I hope to see you guys next week. And if you guys have any questions, feel free to contact me. Yeah, thank you. I know that this was kind of a longer one, but cellular respiration is kind of a doozy in itself. So, awesome. Great. I'm super happy to hear that. Okay, guys, I will see you next week, and have a really nice evening, an awesome Halloween. Please have a fun Halloween for me. Okay. Thank you. Bye.